So welcome back. Now that we're done with cyclic AMP as a second messenger system, let's learn about a second one which plays a role in many different hormones. And this is one that's going to involve an enzyme called phospholipase C. So this is an enzyme which is on the plasma membrane of a cell, and it's going to be activated by a G protein. And what this enzyme does is it acts on this compound right here, abbreviated PIP2, which is phosphatidyl ionocetal biphosphate. Okay, so this is the phosphatidyl part, and this is the ionocetal, and there are two phosphate groups. And you don't have to know this, but basically this PIP2. This is a peripheral protein located on the inner part of the phospholipid bilayer. And so phospholipase C actually cleaves it into right here, so that 3 phosphate groups stay attached to this part, and then this part, which are the fatty acid chains, don't have any phosphate attached to it. So it actually produces 2 different second messengers, but I'm going to show you how it produces the 2 and what they do, and then we will talk in more detail about one of them. So the first one it produces is something called diacylglycerol, which you can abbreviate it as DAG, D-A-G. And so that is this glycerol, which is here attached to 2 fatty acid chains. Now this was how the PIP2 was attached to the plasma membrane. So this part, when the cut has been made in PIP2, this part stays attached to the inner membrane. And what DAG is going to do in order to become a second messenger is it's going to activate protein kinases. Okay. Only we're not really talking about DAG when we're talking about hormones. We are more interested in the other part. So the other remnant of PIP2 is this cyclic protein with 3 phosphates. And what happens is, because it's in a watery environment, this bond is going to break. So the phosphate will have 3 oxygens attached to it, and you'll add a hydroxyl group there. And then this is called inositol triphosphate, or IP3, which is what you can call it. And so now this is no longer attached to the inner portion of the plasma membrane, and so it is in the cytosol. And what it's going to happen, it's going to cause calcium to be released into the cytosol. And it's not going to do that by activating any calcium channels on the plasma membrane. Instead, what happens is IP3 diffuses away from its site of formation towards the endoplasmic reticulum, which has IP3 receptors. And when it binds to those receptors, that causes calcium to leave the endoplasmic reticulum where the concentration is high, and to exit into the cytosol where the concentration is lower. So then the cytosolic calcium levels will rise. And this calcium can bind to calmodulin, like we talked about previously when we learned about smooth muscle contraction. So let's look at this system in detail. Here we have our first messenger, our hormone, binding to its transmembrane receptor. Okay, that is the hormone receptor. This is the target cell. When the hormone is bound to it, that is going to activate a G protein. And then the G protein is what activates phospholipase C. Okay, so remember, the G protein always activates a hormone. In this messenger system is phospholipase C. Phospholipase C is now going to act on PIP2, which I think you clearly can see is a peripheral protein on the inner portion of the phospholipid bilayer. It cuts it right here, which leaves DAG still attached, which can act as a second messenger in other pathways we're not concerned about today. The other part becomes IP3, which is acting as a second messenger traveling in the cytosol to the endoplasmic reticulum where there is an IP3 gated calcium channel. When IP3 binds to this, that's going to cause an egress of calcium from the endoplasmic reticulum into the cytoplasm, where then it can act as a second messenger, activating various proteins and causing cellular responses. So take a moment and let's compare and contrast these two systems. Starting first with, on the right, the simpler cyclic AMP method. And the hormones that are dependent on cyclic AMP are numerous. It's the 4 hormones that form the flat of flat pig, which you'll learn about pretty soon, as well as glucagon, the hormones involved in calcium homeostasis, as well as the catecholamines, which are being released from the adrenal medulla. In contrast, the more steps, more complicated PIP2 to forming DAG and IP3 system is involved with a smaller number of hormones, oxytocin being the primary hormone, except for 3 of our releasing hormones from the hypothalamus. Okay. So take a moment, pause this, and talk yourself through both of these processes to make sure you understand it. And thank you, and I'll see you again shortly as we continue our study of the endocrine system.